Hi, this is Miss Litton, and these wonderful AP Biology students are here at lunch reviewing for their first unit exam. Say hi. Hi. All right. So, first things I want to discuss. You have two parts to this test. You have an essay portion and you have a multiple choice test. When you come to class on Tuesday, you will need to have a pencil if you want to have a pencil to work some things out, but you will definitely need to have a blue or black ink pen. Okay? I will give you a piece of paper that has questions on it for your essay part. Okay? You will answer your questions. It is a confined area because I expect if you know the answer to that particular question, you should be able to communicate it to me in that amount of space. And those questions, on the essay questions, you have a source of 3 different areas in order to be prepared for the essay portion of the test. What are the 3 different sources that you have access to? Textbook at the end of each chapter has essay-like questions. Essay and or short answer and or calculating type questions. Okay? Number 2. What's number 2? Yes. There is a PDF. The PDF is like old school questions, but they're like, this is a stapler type question, right? Content-based questions, and I ask those as well. And place number 3 is what?
Your website. Resources for review.
Yeah, resources for review. They're all in there too. But that's a Google Doc, and it asks more AP-like questions. Keep in mind, those are hard to give out initially, because they're big questions and they are across several different chapters of information. You kind of have to stitch it all together. So those are harder to answer until you've got more content under your belt and more science practices that you can use under your belt. But those are there as well as grading. On the essay portion, not only could I ask you things that are content-based that you see right here, but also science practice-based. And if we look at our different science practices that we have, you could have things like modeling. How would you model this? How would you interpret this graph? Can you do this math? Okay, all of those things are things that I could ask you to do. I could just have a graph on there and have you analyze that data for me. I could give you some numbers and have you do a chi-square test. So those are the things you want to be prepared for the written part. Writing neatly, reading the question, and be able to respond within the right amount of time. Half your time is dedicated to the essay, and half your time is dedicated to the multiple choice, because that's how it will be on your AP exam as well. So I will collect everybody's essay. There will be a time limit. You will turn them in. Then we will transition to the multiple choice part of the test. When you take the multiple choice part, you will have 25 questions, right? Is that right? Was it 25? I was trying to remember. I haven't finished writing it yet, so I don't really know. But I usually do 25 questions. There are 2 points apiece, so it's a total of how many questions? 50. Or 50 points. Sorry, 25 questions too. I can do the math. I can do that one. Okay? So you have 50 points there. Now, the multiple choice, you have the same amount of time. You have to think it through. You have to pick your best answer, and then that's it. When I collect it, obviously I'll be able to calculate your multiple choice score right away, but it'll take me some time to read through all your essays. I read them against a rubric. There are certain things I'm looking for in your responses. And I want you, if you make a claim, I want you to support it with evidence. If you use a term in your essays, I want you to define that term so that you and we know we're talking about the same thing. I would say number 1 errors that I see on essays is people not answering the question that I asked. They feel comfortable about a topic that's related to it or a little sidebar of it, and so they write a whole bunch about that, but that's not the question I asked. You need to be able to communicate effectively. Now you have a student right here who took, and she did not know, I'm putting her on the spot. Would you give them any recommendations for studying for their first exam?
Definitely go to the old PDF that you showed us. A lot of the problems are just like that.
Okay, so she gave you a little tip right there. So make sure that you answer those. Okay, how can you practice multiple choice questions? End of the chapter. Take multiple choice questions. Okay? So that when you take them with me, you're not, it's not your first time. Okay? So make sure that you do that. Okay? That is the process of the test. Do you need to ask me any questions about the process of the test or how the test goes down? All right. So now let's talk about topics. So you have these 4 different chapters. When I look through this, let's make a list of like key things I would know, okay?
Lucas Valencia, please come to the counseling office. Lucas Valencia, please come to the counseling office.
So when I look at the first part, there's a whole section just on the characteristics of life. Okay? You would be surprised how many people, if I ask them the characteristics of life, they will actually miss that question. Okay? That is like a T-ball question to me. So make sure that you know that. The second one that we talk about is we start going through the big ideas. And the first big idea is about what? Evolution. So look at some of the information that we have underneath evolution. What are some key words under evolution that you remember?
Natural selection.
Natural selection. Good. And natural selection is the mechanism, right, for evolution? Okay. What's another word that we use?
Adaptation.
Adaptations. I would be familiar with that. Okay. What's a thing we talked about, like the original ancestral species? Luca. Good. All right. What is the second? This was big idea 1. Okay. Big idea 1. Oh my gosh.
Logan Nolan, can you please come to the office? Logan Nolan, can you please come to the
What
office?
was the acronym we used to remember the big ideas? EEIS. Okay, so this is our first E, was evolution. What's our second E? Energy. Okay? What do we have to expend energy on in order to maintain what? Homeostasis. So you would know what homeostasis meant, right? And you could give me maybe an example of that. Okay, and chemical cycling versus energy flow. Hopefully you know that. Okay, EEIS. What was the I? What's big idea 3? Information. What is our code book for life? DNA. Okay, good. You could review those terms underneath there as well. And then what's the S? Systems. Now, under systems, we talked about what? You remember? Did we talk about organization of life? Atom, what? Molecule, organelle, cell. Why did we go like this with cell? Yes. Okay. We also talked about emergent properties and cooperation, and then we finished up this whole unit here on we finished it up on the science practices. Okay, and we went through all of those, and then the big focus is the scientific method. So if I gave you a certain experiment, could you set it up? Could you come up with a hypothesis if I gave you some information? Now, it doesn't mean that your hypothesis is the right one, right? And we talked about research hypothesis and null hypothesis, right? But could you come up with that? What kind of data would you collect in this situation? Could you analyze that data? What is something I gave you to analyze your data? Chi-square test. And this would be where I'd ask you to do the chi-square test. Okay? That's chapter 1. Okay? Then we moved on to chapter 2. And chapter 2 was your basic chemistry. Now, remember on the organization of life right here, how does that start? Atom, then what? Molecule, and then what? Organelle, and then what? Cell. Okay? So right here, atom, this would be your chapter 2. We learned all about atoms, right? We learned about the structure of atoms. We learned about atomic number, atomic mass, and energy levels, how we fill them. We learned about the differences between ions and isotopes, right? And then we talked about why would atoms ever become molecules, and they do that to get what? Stability, right? And what were the 2 ways we could make an atom happy? Octet rule, get its outermost energy level to 8, or get it full. Okay? And that's why atoms interact with others. And we could predict how many bonds they would make, right? Based on how many valence electrons it has. Then we started talking about the different kinds of bonds there are. What are some of the different kinds of bonds we discussed? Polar bonds. Now, there was polar and nonpolar, which falls under the umbrella of what? Covalent bonds. And in covalent bonds, what are you doing? Sharing electrons, right? Whereas ionic bonds, in order to get stable, you are what? Giving or taking, right? You're ditching an electron or gaining an electron, and then all of a sudden you have a what that you're holding? Charge, right? And opposites attract. But then there are other bonds as well. As a result of polar covalent bonds, right? You know how I'm predict what I'm going to say next. You can end up forming hydrogen bonds, right? Why would you do that? Because as a result of polar covalent bonds, somebody has a little bit stronger pull on the electrons. So they're going to be partially negative, and whoever doesn't have the electrons is going to be partially positive. So now you have polarity in a molecule. And when we talked about with that was the significance of water, right? Because when you see that bond, you have oxygen with 2 hydrogens, but who has a stronger pull on the electrons? Yeah, the oxygen does. So as a result, the oxygen becomes partially negative, and the hydrogen becomes partially what? Positive. So when you have several water molecules together, okay, when you have, okay, so when you have several water molecules together, they will arrange themselves, right, so that the partial negatives are attracted to the partial what? Positives. And what is that called? Yeah, one water to another, specifically, that's called what? Cohesion, right? But that kind of bond right here, this is a what bond? Hydrogen bond, where that bond right there is a polar covalent bond. Now, what if this water molecule is attracted to some surface that's polar? What would that be between that surface? Adhesion. It's still a hydrogen bond, but adhesion is when it's attracted to that other surface. All right. And we talked then about water, about all the properties of water. It has a high boiling point because all of those hydrogen bonds can hold all that energy, right? High point of evaporation because it can hold that energy. It helps moderate our temperatures. It fills vessels. It's a good solvent if it's a polar or charged molecule, right? Ice freezes and it's less dense. Those are all properties of water. Why do we care so much about water? Because we're mostly water. Cells are mostly water. Okay? So then that took care of chapter 2, and then we moved into chapter 3. Chapter 3, we learned how monomers can become what? Polymers, or how polymers can become monomers. How do monomers become polymers? Dehydration, synthesis, or another name for it is condensation. What's the other way? Hydrolysis. Then we just had to learn what the monomers were for the polymers. What were our 4 important polymers?
Carbohydrates.
Carbs. What else? Lipids. What else? What? Proteins. And what else? Nucleic acid. And we know the functions of these 4 polymers, right? And we know what each of them are built out of, and we know carbs are ultimately built out of what? Sugar. And I had you learn some different monomers, right? Okay? Glucose, fructose, galactose, right? Okay? And we talked about how carbs can be used for structure, but they can also be used for what? Energy storage. And we talked about how it would be different if you're in a plant or an animal. Lipids was a weird one, right? Because lipids could be broken down into what? Fats and steroids. And a typical fat would have glycerol with long what? Fatty acid chains. But if we replaced one of those fatty acid chains with a phosphate group, we could have a phospholipid. Or we could take a long chain fatty acid connected to an alcohol, and then we have wax, right? Then a steroid was a little bit different because a steroid was made out of what? 4 carbon rings, right? And we talked about an example of a steroid would be a hormone, a chemical messenger in our body. Okay, then we talked about proteins. What are proteins built out of? Amino acids. Do you know the structure of an amino acid? What are some things all amino acids have in common? Central carbon, go up. Amino. R group. How many different R groups are there? 20. So there's 20 different amino acids.
Attention students, come to the Great Lawn and see the Rocket Club, look at their telescope, view the pictures that they took of the eclipse right now in the Great Lawn. Thank you.
Or stay with me. And then the other item, okay, we want to know about those R groups, is some of those R groups could be polar or non-polar. Then a critical part about proteins is when we hook them together through what process? Dehydration synthesis. What's the name of the bond between them? Peptide bond. When I say what is the primary structure, you're going to tell me that is the sequence of amino acids. Primary structure. You could just list them off. You're just listing them, right? Secondary structure, I have 2 choices. What are my 2 choices? Alpha helix or beta pleated sheet. Is that dependent on the R? No. That's dependent on either end, the amino and the carboxyl groups. Yes? Okay, the tertiary structure is the what? Folding pattern, right? Imposed upon the secondary. What does that do to? R groups. And then quaternary structure is if I have what? More than one chain. Okay? So, and then we had all those functions that we have for proteins. Who codes for our proteins? DNA. Okay? And that brings us to nucleic acids. Nucleic acids, we have DNA, RNA, and ATP. Now, DNA, let's talk about DNA. DNA is a double helix where RNA is a single. DNA has the sugar where RNA has the sugar. DNA has the base thymine where RNA has the base. DNA is primarily located in the of what kind of cells? Eukaryotic cells. In a prokaryotic cell, what would we call that? Nucleoid regions, right? Okay. Remember the kind of RNAs we had? Messenger RNA, ribosomal RNA, and tRNA. I'm curious, in a eukaryotic cell, where would our RNA be made? It's concentrated in the where? Nucleolus. Remember inside of the nucleus? Remember that? Okay. We also talked about ATP, adenosine triphosphate, right? And that's adenine with a ribose with 3 phosphate molecules, and ATP is the energy currency of our cell. All right, that was chapter 3. Now, chapter 4. Chapter 4, the biggest thing we did at the beginning was knowing the characteristics of a prokaryotic cell. Hallmark of a prokaryotic cell is it has no what? Nucleus. It is what compared to a eukaryotic cell? Smaller. What does it have that a eukaryotic cell has?
Cell wall.
If a eukaryotic cell has a cell wall, it will have a cell wall. What else? Cell membrane. What else? DNA. What else? Ribosome. Cytoplasm. Okay? Those are all good examples of things prokaryotic and eukaryotic cells have. Prokaryotic cells are found in 2 different domains. What are the 2 different domains that prokaryotic cells are found? Bacteria and archaea. What makes archaea different from bacteria? Their cell walls. Cell membranes are different. Chloroplasts are in eukaryotic cells. Cell walls, cell membranes, and they live in what kind of environments? Harsh, extreme environments. Okay? Then that brings us to eukaryotic cells. For eukaryotic cells, you want to know that plant and animal cells are different. but they're, they have similarities as well. So you want to know what organelles are found only in a plant cell, what organelles are found only in an animal cell. And then you just need to be able to recognize each of the organelles and what they do, structure and function. If I say powerhouse of the cell, you're going to tell me. If I'm going to tell you photosynthesis, you're going to tell me. And then you're going to tell me mitochondria and chloroplasts have something in common. They both have double membrane, which means they could have been in gold. They both have their own DNA. And that DNA is in a circle. Plasmids are additional DNA found in prokaryotic cells and sometimes eukaryotic cells, but it's circular, right? Who else has circular chromosomes? Bacteria. They have their own ribosomes, and they have their own enzymes, and they are self-replicating. In fact, mitochondria and chloroplasts might have been free-living bacteria, prokaryotes going to live in another prokaryote in order to make a eukaryotic cell, and that is referred to as the symbiotic hypothesis. Okay? We know the pathway a la protein, right? The code starts in the nucleus. We take a message out from the nucleus in what? Messenger RNA. Messenger RNA gets on a workbench known as that ribosome might be found on the ER, or it could be out in the cytoplasm. If it's found on the ER, when that protein is synthesized, that protein's probably going to move into the interior of the rough ER. Okay? So it'll move, good to know, because you missed that part right there. It'll move into the rough ER, then it can be packaged and then go to the Golgi, or it might move into the smooth ER as well. Okay? But then it goes from the Golgi. Ooh, what part does it approach the Golgi from? What side? Cis. And then it'll move out the trans. Then it might go up to the cell membrane and go bleh. Or if it's the stomach of the cell, it might be a lysosome, which will hook up with an endocytotic vesicles to do some digestion, right? What would be a good way for me to take care of poisons in my body? Use my smooth ER could help me do that. What else? Peroxisomes to remove the hydrogens and give it to oxygen in order to form hydrogen peroxide, right? Who is the UPS man of my cell? Who decides who goes in and out? So do you think if I said to you as a question, name the 5 key organelles. I'm giving you a choice. You could pick 5 key organelles. Tell me what they do and support to me why you think they're the 5 key organelles. Okay, nucleus might be a key one. What would be another one?
Cell membrane.
I'd probably say a cell membrane. You better have a barrier. Do all cells have cell walls? No, so I wouldn't pick that as one of my 5. Mitochondria, why? Yeah, you gotta have energy. What else did you pick? Ribosomes. Now, is a ribosome an organelle? No. So now think again. What else could you say? Rough ER that has ribosomes on it, because that's where you might be doing some protein synthesis. Do you see what I'm saying? You want to be able to justify. Here I am for this short amount of time. You want to be able to justify your reasoning of why you think it is the most important structure, right? Just don't go laying it out there and go, lysosome. Okay. Does every cell have a lysosome? No. Yeah, I'm sure you could justify you need nutrients, but plant cells do fine without them. So I wouldn't necessarily pick that. I'm looking for you to be able to evaluate and decide and support your claim of these are the 5 most important organelles and why. I'm looking for you, if I ask you, what do you think are the most critical properties of water? Are you gonna tell me, oh, water strider, needs to be able to go, that's super important, You're right, for a water strider, okay? But maybe not all along. So you want to think, what would be a critical? Know it well enough that you can evaluate the most important. Okay, and I think, my friends, that's kind of all it. Write your essays, take multiple choice tests. okay, in order to get ready. Any other questions you want to ask me while I'm here? Is it important to have fun this 3-day weekend?
Yes, of course.
Damn straight it's important, okay? So listen to me. Listen to me. Hear me. Guys, your number one problem is going to be procrastination, except for some of you who need counseling. Your number one problem is going to be procrastination. Here is my strong suggestion to you. Give me at least 30 minutes today where you review. Review something today for 30 minutes. You know what the hardest part of having a major review or a project is? Getting started. Start something today. Find some time tomorrow. Look at something. All of you have things on your phone. So even if you're going to spend the day at Disneyland, I bet you're waiting in line at some point. Bore your parents to death. Go, I'll tell you about a cell membrane, okay? And then give some explanation. Visit a little bit every day. I do not want you Monday night freaking out about your test on Tuesday. Do you hear me? No freaking out. No mental breakdowns. I love you very much. Make good choices.